Hello everyone. Today we are going to explore a fundamental aspect of microbiology, microbial culture. Microorganisms are everywhere. In the air we breathe, the water we drink, and even within our own bodies. But to study these teeny life forms closely, we need to grow them in controlled environments. This process is known as microbial culture. In simple terms, microbial culture is the method of multiplying microorganisms by letting them reproduce in a specific environment under controlled conditions. This technique allows scientists, doctors, and researchers to identify microbes, understand their behavior, and even develop treatments. In this video, we will look at different types of microbial cultures, from basic methods used in the classrooms to advanced techniques applied in clinical labs. Let's begin by understanding why microbial cultures are so important. A microbiological culture media is a source of nutrients to support the growth of microorganisms in vitro. This media helps in the growth and counting of microbial cells, selection of microorganisms, and survival of the microorganisms. The first artificial culture medium was created by Louis Pasteur in 1860. Culture medium is essentially composed of basic elements, water, nutrients, to which must be added different growth factors that will be specific to each bacterium and necessary for their growth. Now, what are the common ingredients in culture media? Peptones and proteins, which are source of nitrogen. Yeast extract or mist extract, which provides vitamins and growth factors. Carbohydrates, these carbohydrates are the energy source, and the examples of the carbohydrates are glucose, lactose, etc. Agar, which is a solidifying agent. Salt and buffers. The salt and buffers maintain the osmotic balance and pH. These are the ingredients of media. Now let's discuss about the types of culture media. Culture media are categorized based on their physical nature, chemical composition, and also on functional type. Based on the physical nature, they may be either liquid or semi-solid or solid. Based on the chemical composition, the culture media are synthetic or defined media or complex media. Whereas based on functional type, the culture media are basal media, enriched media, selective media, differential media, and transport media. Now, based on the physical nature, we have already discussed that these are liquid media, semi-solid media, and solid media. Now, let's discuss about liquid culture media. This liquid culture media, also known as culture broths, where nutrients are dissolved in the water. These are nutrient-rich solutions used to cultivate microorganisms in a liquid state. As it is liquid culture media, so here microorganisms are cultivated in liquid state. This liquid culture media facilitate faster growth, higher isolation rates compared to the solid media. Commonly used in fermentation studies, allowing for the large-scale growth of microorganisms. This liquid culture media are used in fermentation studies because this liquid culture media facilitated faster growth and higher isolation rates. Thus, it allows for large-scale growth of microorganisms. Examples of liquid culture media are nutrient broth, tryptic soya broth, various specialized broths for specific bacterial species or applications. Next, what are the applications of liquid culture media? Liquid culture media are used in various purposes. For example, these are used in research, in industrial process, and in diagnostic testing. These liquid culture media are used in research to investigate microbial growth, metabolism, and various biological processes. In industrial process, in industrial fermentation process for producing various products such as antibiotic, enzymes, biofuels. For diagnostic testing, it is used in clinical laboratories for identifying and analyzing microorganisms which are present in patient samples. Next is semi-solid culture media. The semi-solid culture media containing 0.2 to 0.5% agar and are used to demonstrate bacterial motility, cultivate microaerophilic bacteria, and appearing as a soft jelly-like substance. So as it is semi-solid culture media, so it appear as a soft jelly-like substance. And here the solidifying agent is agar, which concentration is 0.2 to 0.5%. Okay, this is very less. And it used to demonstrate bacterial motility and cultivate microaerophilic bacteria. Microaerophilic bacteria are those which require oxygen but in very low concentration in contrast to aerobic bacteria and anaerobic bacteria. In aerobic bacteria, they does not require oxygen and they will die in the presence of oxygen, whereas the aerobic bacteria require oxygens for their growth and survival. Examples of semi-solid culture media are mannitol motility medium and Stewart's transport medium. Next, now what are the applications of semi-solid culture media? Semi-solid culture media used for determining the bacterial motility. These are useful for determining bacterial motility as motile bacteria can move through the medium while non-motile bacteria remain at the point. Cultivation of microaerophilic bacteria. These are used for the cultivation of microaerophilic bacteria which require low oxygen environment. Semi-solid culture media also used for transfer of cultures and also used to identify characteristics of microorganisms. So these are the common applications of semi-solid culture media. Next is solid culture media. Solid culture media essential for culturing microbes uses a solidifying agent like agar to create a surface where individual colonies can grow and be isolated. Here the agar concentration is 1.5% to 2%, whereas in semi-solid culture media the concentration was 0.2 to 0.5%. Okay. Examples of solid culture media are nutrient agar, blood agar, McConkey agar and chocolate agar. Now what are the types of solid culture media? There are three types of solid culture media mainly. One is agar plate, second one is agar dipped and the third one is agar slant. The agar plates. Here solid media poured into petri dish and allowed to cool and solidify. It allows for easy colony growth and observation. Application. These agar plates are used for isolation of colonies. Next is agar slant. Here solid media solidified at an angle in test tube, providing larger surface area for growth. Now application. These agar slants used for long-term storage of cultures. That is how long? Few weeks to several months. Next is agar dip. Here solid media solidified upright in test tubes and used for culturing bacteria that require deeper growth. Application. This agar dip is determine bacterial motility and oxygen usage of a culture. Now what are the applications of solid culture media? Solid culture media are used for isolation of pure cultures, for identifying colony morphology and also for streak plate method. For isolation of pure cultures, solid media allows for the separation of individual bacterial colonies, each originating from a single cells, enabling isolation of pure cultures. Next is colony morphology. The solid culture media are used to identify colony morphology. The characteristic of colonies, that is size, shape, color, texture, can be used to identify different bacterial species. The size, shape and color and textures are vary in different bacterial species. Thus, you can identify different bacterial species. Next is streak plate method. The common technique used to isolate colonies on a solid media is the streak plate method, where sample is streaked across the surface of the agar to create individual colonies. Now, culture media are also categorized based on their chemical composition. One is synthetic or defined, and the second one is the complex media. Now, let's discuss about synthetic or defined medium. Definition of synthetic or defined medium is that a chemically defined or synthetic medium is a growth medium where all chemical components are known and there are no undefined ingredients like yeast extract or animal derived products. So here all chemical components are known, that's why it is defined medium. Now, what is the purpose of using this synthetic or defined media? These media are used for precise control over the growth environment, allowing researchers to study the effects of specific chemicals on cells or microorganisms. Next is composition of synthetic or defined medium. Defined media contain pure known chemicals such as salts, sugar, amino acids and vitamins, and in specific concentration. So in synthetic or defined media, not only all the chemical components are known, but their concentration are also specific. Examples: Dubose culture medium with 280. Now, what are the applications of this synthetic or defined medium? Precise control. Second one is reproducibility. And third one is the elimination of variability. These are the application of defined or synthetic medium. Researchers can precisely control the composition of the medium, which is essential for studying specific metabolic pathways or nutritional requirements. Second is reproducibility. The known composition ensures that the medium can be replicated consistently, leading to more reliable results. Third one is elimination of variability. By using pure and known components, the variability associated with undefined media such as batch to batch differences in complex media is reduced. Next is complex media. The complex media, also known as undefined media, in contrast to defined media, that is defined media or synthetic media. So complex media, also known as undefined media, are microbial growth media containing a variety of nutrients from cell extracts like yeast, meat, plant sources, where the exact composition is unknown, allowing for diverse microbial growth. So in complex medium, the exact composition is unknown, whereas in synthetic or defined media, the exact composition is known. The nutrient source. The complex media contain rich nutrient source, including water-soluble extracts of plants or animal tissue such as peptone or tryptone, enzymatically digested animal protein. So this peptone or tryptone basically are enzymatically digested animal protein. Now examples. Common examples include nutrient broth, nutrient agar, tryptic soya agar. Now, what are the advantages and disadvantages of complex media or undefined media? The advantages include versatility and easier preparation. So, versatility. Complex media can support the growth of a wide range of microorganisms due to their diverse nutrient content. Next is easier preparation. They are relatively easy to prepare as the exact composition of the ingredients is not critical. That's why these are easy to prepare. Not disadvantages. Disadvantages is lack of precision. The unknown composition makes it difficult to control the growth of specific microorganisms or to study their metabolic pathways in a precise manner. This is the disadvantage of complex media. Now, the media are also categorized based on their functional type. So, based on the functional type, the media may be basal media, enriched media, selective media, differential media, and transport media. Now let's discuss about basal media. These are simple general purpose media that support the growth of many non-fastidious microorganisms. So what is non-fastidious microorganism? Non-fastidious microorganism are those that have no specific nutritional or environmental requirements to grow and reproduce. Examples include nutrient broth or agar and peptone water. Next is enriched media. These media contain additional nutrients or growth factors to support the growth of more demanding fastidious microorganisms. Now, these fastidious microorganisms are those that have specific nutritional and environmental requirement. Okay, to grow and reproduce. Examples are blood agar. Blood agar is enriched with blood which provides growth factors and nutrients of fastidious bacteria such as Haemophilus influenza. Second example is chocolate agar. It is an enriched medium because it contains added nutrients primarily from the heated blood which are essential for the growth of fastidious bacteria like Haemophilus and Neisseria species that have specific nutritional requirement. Next one is selective media. These media contain ingredients that inhibit the growth of certain microorganisms while allowing others to grow. Examples include mannitol salt agar, which is a selective for Staphylococcus species, which can live in areas of high salt concentration. Next is differential media. Differential media allow for the visual distinction of bacteria based on their biochemical properties, showing different growth characteristics or colony colors on the same plate. Examples: eosin methylene blue, that is EMB agar. These are used for identification of gram-negative bacteria, especially Enterobacteriaceae. EMB inhibits the growth of most gram-positive bacteria. Next is McConkey agar. It is differential for identifying lactose fermenting from non-lactose fermenting gram-negative bacteria. It also selects gram-negative bacteria by inhibiting the growth of gram-positive bacteria. Next is transport media. This media are designated to preserve the bacteria during transit to the laboratory without allowing specific growth. This media often contains specific buffering agents, reducing agents to maintain bacterial viability. So, basically, this media helps in maintaining bacterial viability while it reduces significant growth of the microorganisms. Examples include cadmium medium for stool or rectal swab transport, viral transport medium or VTM for nasopharyngeal swab transport. So in short, microbial culture media allows for the isolation, identification, enumeration and functional characterization of microorganisms from a wide variety of environments including clinical, environmental and industrial samples. Different types of culture media are available based on their physical nature, chemical composition and functional type. We have already discussed and we can use them based on our needs. Thank you. If you like this video, please like, share and subscribe to this channel.